when I was at Caltech. And then when we were considering coming to Germany, again, we heard a lot about the fabulous group working with human patients here and working on memory mechanisms. And not being well acquainted with German geography, for me it was just fabulous to get on the train and be here in 2 hours and sort of see this place on the map and see how easy it is to get here and to begin to meet what I hope will be some new colleagues. So my lab is interested in information storage in the brain, and we study this at many different levels, ranging from looking at protein trafficking and messenger RNAs present in the dendrites of neurons and trying to understand the cell biology of how information is stored at synapses. But we're also interested in how information is stored in circuits, and became interested also in studying this in humans when the opportunity arose very early on in my time at Caltech. And actually, as an undergraduate, I studied human memory when I was at USC. My honors thesis was on basically doing tests of human memory in twins. So I'm not completely crazy to be interested in using humans also as subjects for memory studies. So I'll try to not bore the aficionados here, but also provide some background. This is a map of some convenient nomenclature that has been used to describe human memory, basically declarative memories, which would include episodic memories or semantic memories, versus procedural memories, which are more sort of automatic kinds of learning that include simple classical conditioning and skill learning, like riding a bicycle, and effects like priming. And in the studies that we've done in human subjects, we've been particularly interested in focusing on episodic memory because of some features. One is that is particularly nice for if you think about tasks that you can use in a clinical setting. It's desirable, if you want to look at all stages of learning and memory, to have something that doesn't take a lot of trials. And so something like an episodic memory, which can be learned in a single trial. They're often records of events, egocentric, and possess many attributes, which is, of course, what one of the things we know about the hippocampus is that it receives information from every sensory modality. And so if one is interested in what the hippocampus does for information storage, it's good to look at memories that have many different aspects. And then also nice is that in principle, for some episodic memories, they can, even though they can be induced by a single trial, they can be remembered for a lifetime. So if you want to understand the basis for long-term information storage in the nervous system, this is a particularly nice system. And we have focused our studies on the hippocampal formation and other brain structures that are in the medial temporal lobe. And most of the experiments that we've done and that I'll describe today were done in the hippocampus. And in the human studies, we have done single unit recordings from hippocampal neurons and amygdala neurons primarily. Now, we, like all the other groups that are interested in using this technology, have been fortunate to, in some ways, take advantage of a very special population of epileptic patients. Those are patients that whose epilepsy is not treated by medication, and furthermore, whose epilepsy, they become candidates then for resective surgery. And their epilepsy, the seizure focus cannot be defined by scalp EEG electrodes or grid electrodes, so they become then candidates for the implantation of depth electrodes, which have EEG contacts on them. And these are implanted into various medial temporal lobe structures. These are the EEG contacts, and I'm sorry this is so dark with this projector, but this is through this what we call macro electrode that records the LFP. We can thread a bundle of microwires through this, like as if this were a cannula. And at the tip of this macroelectrode then emerges this sort of flower of wires, which are capable then of recording single unit activity. So the action potentials that come from individual neurons. And these electrodes, in each macroelectrode, we have 8 microwires. They're platinum iridium wires, 40 microns in diameter. And within this spray, we typically use one of the wires as a ground. And so we have 7 wires that we can record individual unit activity from. We now is, the group that I've primarily worked with is Uli Ruteshauser, who was a graduate student and then a postdoc in my lab, and he's actually responsible for all of the recordings and data analysis that I'll tell you about. And in the early studies, we worked together with Adam Mamalek, the neurosurgeon, who was doing the depth electrode implantations at the Huntington Memorial Hospital, which is right next to Caltech. And Adam has recently relocated to Cedars-Sinai on the west side. So we've been working with 2 other neurosurgeons now at Huntington Hospital, Ian Ross and Chris Heller. So the electrodes are implanted according to previous clinical criteria using stereotactic devices and the software that allows one to actually look at computerized images of the patient's brain. And so we have no say in where the electrodes go, but just take advantage of those electrodes that are placed in the medial temporal lobe. And this just shows some examples of the electrode locations. The green, these are 2 different patients with different MRI images showing the electrodes. And it might actually be helpful if the lights could be down a little bit, just because I'm seeing that a lot of the images aren't as bright as they could be. Okay, so now you can see here, here are the eyes, the back of the brain. You can see the electrode coming in here. This is the amygdala electrode, hippocampal electrode. The electrode, of course, looks bigger than it really is. This just shows the contacts, and this is the region where the little spray of microwires comes out. Now we record activity on all the microwires, and often we have as many as 8 macroelectrodes. That would be a lot. And so we can record, this is the raw signal that we record, and we can record then individual spikes that we can resolve and attribute to the activity of a single cell by using typical kinds of spike sorting technologies, where you end up with a cluster of units. And this is an example of one unit that we isolated. This is the waveform, so this shows the overlay of 26,000 spikes that we recorded that we attribute to this single cell. And then we do a variety of tests to ascertain whether this is indeed, these spikes arise from a single cell. And one of those is to look at the interspike interval, which of course, if it's coming from a single cell, the biophysics of action potential firing put limits on how rapidly you can detect 2 spikes. And we can see here that a very, very, very small fraction of our spikes detected, of these 26,000 spikes, are possess an ISI that's not possible, basically below 3 milliseconds. So this tells us that the majority of these, an overwhelming majority of these spikes are likely to emerge from this single cell. We've also looked carefully at the firing rates of the cells and have characterized the firing rates in the right hippocampus, left hippocampus, the amygdala. And we've also looked to see for the particular tasks that we are looking at, so these would, and I'll show you an example of that in a minute, look to see if the cells that we are selecting for our analysis differ from the cells that we do not choose because they're not responsive, and we find that there's no significant difference in the firing rate between these 2 populations. So we're not selecting some sort of atypical set of cells to analyze. So for these experiments, I'd like to tell you about 2 sets of studies that we've done. One has to do with whether the activity of individual cells actually contains information about whether an image has been seen before. So these would be cells that would indicate that a person has had an experience with a particular image. And the second story I'd like to tell you about has to do not just with the firing rate of cells, but actually about the timing of spikes, which is something that I think is particularly a nice kind of experiment that really exploits humans exploits, hopefully not in a bad way, but exploits the ability to use humans because they can, of course, learn rapidly and report quite rapidly on what they've learned. And in doing so, we can actually measure the importance of spike timing relative to the ongoing population activity and ask whether there's a relationship between these events, between the timing of individual spikes and the local field potential that gives a clue about the successful coding of information in memory. So the basic task that we have used is always a variant of this kind of task, where we have a series of images that are usually natural scenes or objects that are presented either in these early studies in one quadrant or sometimes in the middle. And the task of the subject, they're instructed to look at these images and try to remember them. And then to control for attention, we ask them, in some cases, where was the image when the image is presented in one of these four quadrants? Or in experiments where the image is presented in the center of the screen, we ask them whether they saw an animal or not. So in the learning trial, depending on the capacity, the previously determined capacity of the patient, we will present between 50 and 100 images that we presume are new, are novel to the patient, presume that they have never seen them before. They're images that we have usually gotten off the web. And then with some delay, either 30 minutes or sometimes a 24-hour delay, we do what we call the recognition phase, where a subset of the images that the subject saw in the learning trials are represented. And all the subject is asked is whether they have seen this image before. So they have to press old if they saw it during the learning trial, or press new if they didn't see it. And so the question is, we can look at the memory performance of the subjects, but we are curious to see what neurons in the medial temporal lobe were doing when subjects were doing this behavioral task. And what we found was that we could identify at least two different classes of cells, and one class of cells we called familiarity detectors. So these are neurons that, based on the single exposure to the stimulus during the learning phase, in the recognition phase, signaled that they had seen the image before by an increase in firing rate. So this is now a series of raster plots for one such cell. The gray here represents the baseline period, and here are 36 different images, or actually maybe 40 images that were presented here. And the stimulus was presented at this time, and we've grouped the images according to whether this is the image that was presented during the learning trial or during the recognition trial, and during the recognition trial, whether it was an old or familiar stimulus or a new stimulus. And so the hallmark feature of these familiarity detectors is that they increase their firing rate upon the second exposure to the stimulus. So the cell had a certain baseline firing rate for these 12 different images. But when the image came on the screen here, the cell increased its firing rate, indicating that the image had been seen before, more or less. And the average of the spikes per second are shown here. So when the first time the image was presented, either in the learning trial for one set of stimuli shown here, or during the recognition phase when we presented the new or distractor stimuli, there was no change in firing rate. But when the image was presented the second time, for this one class of neurons, we could see this enhanced firing rate. And we also saw a class of cells that we called novelty detectors, and these are cells that fired only upon the first presentation of the stimulus. So by definition, during the learning phase, the first time the stimulus is presented is the first presentation, and these cells this is now another individual cell showing 35 different stimuli. The 12 stimuli here were the first presentation of a stimulus during the learning trial. And so you can see the baseline and stimulus on here. There was an increase in the firing rate of these cells. And when one looks in recognition, those same stimuli, when they were represented, now no longer elicit that increase in firing rate because the stimuli are no longer new. But the new stimuli that are added as distractors during the recognition phase now also cause this increase. So what these neurons appear to be signaling is the very first presentation of a stimulus. And these, of course, are potentially very important cells because we know that novelty, the stimulus novelty, is an important determinant for whether something is going to be remembered or not, or is able to partake in an associative memory. So the population averages for these two groups of cells are shown here. So here are the novelty cells. So this is the group average showing the increase in firing rate both in the learning phase for the first set of stimuli, and then also in the recognition phase for the distractors that are presented. And when the stimuli are represented, we see actually, interestingly, a decrease in firing rate, this significance, which means that these cells are also convey information about whether a stimulus has been seen before, but they do that by decreasing their firing rate. So there's also some aspect of the coding of these cells that tells that could potentially contribute to object familiarity. And by contrast, the familiarity cells only respond to the stimuli on the second set. So those are the familiar stimuli that are presented during the recognition phase of the trial. Now, we have looked to see whether this kind of property represents long-term memory. And the way that we've done that is to, we can't, with any degree of confidence, keep or say for certain that we are able to hold the same cells for 24 hours, but we can do a population analysis where we can examine the increase in the number of spikes detected in these either novelty or familiarity neurons at 24 hours and 30 minutes. And we can see that as a whole, the population is capable of signaling this kind of information at 24 hours. And this is also true if we compare just the nature of the stimulus, whether it's an old or new stimulus. So we believe that this change that we can detect in these cells represents or stays there in the circuit for at least 24 hours. Now, another thing that we can examine is how much information is actually contained in the firing pattern of these cells. And to do this, what Yuli did was to develop a population decoder, which has information about the number of spikes that are fired in this window of time when the stimulus is on. And he uses a regularized least square classifier to basically pull out of this system how much information there is. What we were interested in is the redundancy of information. So we find these classes of cells, and we were interested in whether the classes of cells are working together to provide more information to the system. And the way that Yuli addressed this was to look and see how many trials based on these spike numbers could be correctly predicted. So is this a new or an old trial? When he added an increasing number of neurons to the overall information. And what he found was that as he increased the number of neurons he used to classify the stimulus as either new or old, the prediction of the model got better and better, suggesting that each neuron is at least contributing something unique to the overall information that the system possesses. And if we did experiments where he scrambled the spikes here, he found that the performance of the decoder was just at chance level. So one interesting feature of these experiments, of course, is not just the trials that the subjects get right, but also the trials where they make mistakes. And in particular, because we have direct access to single unit activity. And we know that, for example, that a population declassifier making use of that brain information can predict with very high accuracy whether a stimulus is new or old. We can ask ourselves, in particular, on the trials where the subject makes a mistake, or their behavior tells a mistake, what was the brain actually coding for on those trials? And so we took a look at the error trials, so the trials where the subject said that, let's say, a stimulus was new when in fact it was old. And we asked for those particular trials, what does the decoder predict? So the decoder that has access to the single unit activity, what does it predict for the stimulus? And there's three possibilities. One is that the decoder could just perform at chance. Half of the trials get the right thing and half of the trials get the wrong thing. Or the decoder could report what the subject did. So in some ways you would argue then that if these brain cells were directly connected to the behavioral output of the cell, then these would be essentially the same thing. So the subject's behavior was just sort of phenocopying what the information in the system was telling it, which was wrong, of course. They said new when the stimulus was old. Or the third possibility would be that the neurons would actually contain the information that's the correct answer. And so what we found, surprisingly, on these error trials was that the error trials, by and large, significantly predicted the correct response, which means that a naive observer who had access to that same brain information would make the correct judgment about the stimulus, even though the subject themselves made the incorrect judgment, which implies either that the nature of the misinformation is somewhere downstream of where we're recording, or that some of the cells that we aren't recording from and don't have access to are contributing to the failure of the subject to deliver the correct response. So now I'd like to discuss a second study that we've done, which gets at this relationship between the timing of individual spikes and memory performance. And I think that this first study that I described, where we're actually looking at rates, spike rates, and just counting spikes in the baseline period and during the stimulus presentation, is it's a good sort of early endeavor to look at rather gross coding features of these neurons. But I firmly believe, like I'm sure all of you or many of you do, that the real perceptual coding in the nervous system is not just done by a rate code, but rather has to do with the timing of spikes, because there's a lot more information rather than a spike or not a spike when one considers the fact that these neurons exist in integrated networks that are changed by activity states and changed by the presence or absence of different neuromodulators. And in this study, we took advantage of the fact that one can record not only individual units, but also record local field potentials from cells. And we were intrigued by some work in the literature that showed, in particular, in looking at plasticity, for example, in the hippocampus, and looking at one prominent form of plasticity where synapses are strengthened, long-term potentiation, that the delivery of stimuli relative to the phase of an oscillation that is recorded with an extracellular electrode, and this is an oscillation that occurs at the theta frequency between, everyone's definition is different, but let's say between 3 and 8 hertz or 9 hertz, that the timing of these spikes relative to the theta frequency predicted whether one would actually be able to elicit long-term potentiation in these studies. So when the burst was delivered on the positive phase of a theta wave, on the up phase, one could successfully elicit LTP, but if the burst was delivered on the negative phase, either 3 bursts or 10 bursts, there was no change in synaptic transmission. So this is just kind of a hint that the timing of spikes is important for information storage potentially in the nervous system. And it's also known that novelty, as I mentioned, is not only important for animal learning, but can enhance the strength and the duration of LTP. So, and novelty influences brain oscillations. And in this experiment, what they showed is that if animals in vivo LTP experiments, if animals are placed in a novel home cage, that when LTP is elicited, the magnitude of LTP is greater, and it lasts actually for a longer period of time than when they're in their home cage. And interestingly, this effect of novelty here is blocked if dopamine antagonists are infused. So we were interested, like many other groups, in examining whether we could detect different kinds of brain oscillations in our human recordings. And these are some examples of raw LFPs that we recorded. And this is after filtering in the theta band or in gamma band. And we could clearly see evidence in these recordings for theta oscillations and also for gamma oscillations. We could see this not only in the hippocampus, but we can also see it in some of our recordings from the amygdala. And we find actually that our theta oscillations are generally slower than what have been reported for other species. So what we then wanted to do was to ask whether there was a systematic relationship between these oscillations, the theta oscillations that we could record, and the timing of the individual spikes that we could record. So here's an example of 2 units that we isolated from this new set of recordings, again showing the overlay of the single unit activity and also the interspike interval histograms for these cells. And what this shows now is the raw trace and the theta frequency trace. And what you can see superimposed here is the spikes of an individual cell. And if you look very carefully, what you can see is that the spikes do not occur randomly along this trace, but rather occur with some preference at a particular phase of the theta frequency. And this is this cell here where we do a polar plot to look at the frequency of spikes with respect to the entire cycle of the theta oscillation. And what we can find is that this cell prefers to fire at a particular phase of the theta oscillation. Shown here, this is at the trough of theta. And this shows the significance of the phase locking of this cell, showing that it shows a significant phase locking in the theta frequency here. Of all the cells that we recorded from, we found about 20% of the cells, and this is a data set of around 300. 20% of the cells are phase locked to an LFP oscillation in the theta range. And we looked at the preferred phase, and what we found is that most cells preferred the 0 phase of either the positive or negative phase of this theta frequency. Now, this task what we did was to record using a very similar task, and we added here a confidence measurement. So during the retrieval time, when the subject is represented with some of the old stimuli that they were presented during learning, as well as a new set of stimuli, they had to answer whether the image was old or new. But now, instead of just new or old, they were asked to say new and confident, new probably, new guess, all the way to old confident. This way we could assess memory strength and relate it to the phase locking of these individual cells. And we analyzed the phase locking during this entire period, during the pre-stimulus period, the time that the image was on, and the post-stimulus period. And this just shows the confidence rating, so it shows that this works, so that the old stimuli, the percentage of responses for old confident are here, and for the new stimuli, the percentage of responses for new confident are here. So you can see that the subjects use this scale effectively. So during during the recognition, we have essentially two sets of trials. So we have the stimuli that the subject remembered, which are true positives, and the stimuli that the subject forgot, which are false negatives. So what we have done is to take the stimuli now in recognition and divide them into these two classes and retrospectively go back and look at the data during the learning trial and ask, is there anything about the synchrony of those individual spikes that we can record relative to the local field potential that predicts the subsequent memory performance that we record in the recognition phase. So we can examine these two types of trials and look at the synchrony. Now, many of the neurons, as you would imagine based on the data that I showed you in the first part of the talk, many of the neurons respond to the stimuli with an increase in firing rate. So here is one particular neuron where you can see 100 stimuli presented, and on average you can see that for many of these stimuli, compared to the baseline period, this cell exhibits an increase in firing rate. And this is the average increase in firing rate with these 100 different stimuli that are presented. But if you divide the trials during recognition, if you take these stimuli now and you ask which stimuli did the subject remember and which did they forget, you can see that the increase in firing rate here had no predictive value for whether the subject was going to remember or not. So regardless of whether they remembered or forgot, the increase in firing rate was the same. So the rate is not distinguishing subsequent memory or not. And this is just to reemphasize this point that there are basically two pieces of information. Every time you look at a spike, you can tell the time that the cell is spiking, so when the stimulus is on or when it's not, but also its phase relative to any ongoing oscillation. And in this particular experiment, we've looked at theta. So this shows how are we going to actually analyze the data. So what we'd like to do is to take the cells, and every time the cell fires an action potential, we capture the local field potential in the negative and positive time direction to look and see what the average of that field potential looks like. And if there's a systematic relationship between the spiking of an individual cell and the field potential, you will see an oscillation in the field potential, even though it's an average. And so this shows for all spikes that this cell fired, shows the average field potential. So you can see the theta oscillation here present in this case. But if we take the same neuron now and we divide these trials shown in green into the two blocks, the remembered trials versus the forgotten trials, what you can see is that the frequency of the spike-triggered average for the remembered trials, this is much higher. The amplitude of the spike-triggered average is much higher for the remembered trials versus the forgotten trials. And this is something that we saw consistently across neurons, that when we compared the remembered versus forgotten trials, we were able to see the amplitude of the spike-triggered average was much greater for the remembered trials than the forgotten trials. So we've looked at other kinds of statistics that can help us to actually quantify this, and these statistics actually normalize for changes in spike rate as well as changes in theta power. And we start with this spike-triggered average, and this is actually work that was done by Pascal Fries, using similar analyses in the visual cortex, looking at two different kinds of visual stimuli. And he was able to take the spike-triggered average for these two conditions and then compare them to show that there was greater synchrony in one condition when compared to the other. And then by normalizing this spike-triggered average over something called the spike-triggered power, you can come up with a statistic that tells you the degree of synchrony and is basically scale invariant. So it's normalized for spike rate changes as well as power changes. So we've calculated the spike field coherence across all cells, looking at the frequencies and across many frequencies to look and see where the coherence emerges. And what we find is that the spike field coherence for remembered trials is cells are much more likely to signal this synchrony in this theta frequency. So the higher this spike field coherence, the more accurately the spikes are following a particular phase of the theta wave. And because we have these confidence intervals, then we can actually do other kinds of comparisons. So we can calculate spike field coherence for trials where the subject had either high or low confidence that they had remembered the stimulus. And what we can see is that even the confidence, the sort of memory strength, you might argue, is represented by differences in this spike field coherence. And this is the spike field coherence for false negative trials, for forgotten trials, for high confidence trials, and for low confidence trials. So you can see that the memory strength is represented here. And this is a similar kind of average where we've now compared the low confidence trials and subtracted the false negatives. And you can still see that there's even a significant relationship for those trials where the subject wasn't 100% sure that they were right or not. So these data indicate then that it's not just the rate of the cells, but actually the synchrony of the cells to the ongoing oscillation. Now we looked in particular at when the synchrony was important, and we did this by subtracting, either doing a null distribution, where we scrambled the spikes relative to the theta, or the real distribution where the SFC is recorded on a heat map here. And these bars here indicate the stimulus onset, and here is the frequency from 2 to 12. So what you can see here is a period of high significance, so high phase locking, in the time when the stimulus is on, in this range of 1 to, let's say, 5 hertz or so. And actually, this area here that's outlined in black is statistically significant. But we also saw an interesting thing that we could see even before the stimulus came on, that there was a period of synchrony during the baseline, which hints at other processes that are not just stimulus-driven, that might be something about attention or arousal. So in none of these experiments can we say what actually the mechanism is. We can just say what we see here. And this shows the periods of significance here, the bootstrap significance test, showing that there was a period of synchrony preceding the stimulus onset, and also a significant period of synchrony during the stimulus presentation that was predictive of later memory success. Now, we were also interested in whether there were changes actually in the theta frequency band itself. And there's a very old study from Richard Thompson's lab looking at eyeblink conditioning in rabbits, showing that the amount and the amplitude of theta that they could record in the hippocampus predict the speed of conditioning. So this is an associative conditioning paradigm where an air puff is paired with a tone. The air puff alone usually elicits a withdrawal response of the nictitating membrane, which is the little membrane that rabbits and other animals have underneath their eyelid. And what they found is that the way that they did the ratio here is the lower the numbers here, the more theta power. That with the more theta power, they had fewer trials to criterion. So trials to criterion is something like when the rabbit learns on 80% of the trials, it shows a conditioned response to the tone alone. And so this very early study highlighted the importance of the baseline theta in potentially influencing the speed of memory. We found that there were no power differences in the theta frequency that we could record when we separated the true positive trials from the false negative trials. Nor were there systematic changes, as I said, in the firing rate of the cells. So when we looked across the entire period here, you can see that for true positive and false negative cells, there's no significant difference in the firing rate. So this suggests then that the enhanced synchrony is not emerging because there's more opportunities for synchrony because the theta power is enhanced, but we rather at least think now that the synchrony may be driven by inputs to the hippocampus that are driving or modulating the activity of the individual cells. So to conclude this part of the talk, and then I just have another short story that I want to tell you. We have shown in single unit recordings from humans that we can detect neurons that respond to new stimuli and familiar stimuli in both the hippocampus and the amygdala. And based in part on the familiarity neurons, we can find cells that respond with a single trial and change their responsiveness. Now, this is something that one could never see, for example, in non-human primate experiments, because as far as I know, most of the conditioning protocols that people use require many, many trials to get to criterion where you can actually see evidence that the animal has learned. So this is, I think, something that is uniquely human, because we have tasks where humans can learn something in a single trial and can actually report to you that they've learned something. So you have a chance to look at very rapid mechanisms for encoding information. And then I showed you that a decoder with access to the neuronal activity sometimes outperforms the behavior of the patients. In those particular examples where we had error trials where the subject had reported the wrong response, when the population decoder looked at the same activity that we could see, it, in 70% of the time, actually predicted the correct response. And then in the second part of the study, I showed you that individual spikes within the hippocampus and amygdala are phase locked to particular phases of a theta frequency wave, and the strength of this phase locking predicts memory strength. I think this has very interesting ramifications potentially for thinking about human learning and memory, because it really points to the importance of network dynamics, and we don't know if the contributions of arousal or attention to these systems, but it clearly shows that these things, in as much as they can influence the synchronous activity of the network in producing theta, potentially could have a very strong influence on how the network encodes the information. Now, in this last little section, I'd like to tell you about some in vitro work that we've done looking at network properties in the hippocampus. And this is just another diagram of information flow into the hippocampus. So information from different polymodal and unimodal association areas goes into the perihippocampal cortices, and then via the entorhinal cortex, information comes into the dentate gyrus, and then the mossy fibers, which are the axons of the dentate granule cells, project to CA3, and the Schaefer collaterals, which are the axons of the CA3 cells, project to CA1, and then the CA1 axons project to the subiculum, and the subiculum feeds back to the entorhinal cortex and also projects elsewhere. So this is the classic trisynaptic circuit that has been studied, I think, more than any other brain circuit. And we became interested, and the nature of information processing as one goes through each of these synapses has been a subject of great interest to many of us. We were interested in another potential source of information that goes directly to area CA1, which involves a direct projection from the entorhinal cortex to the CA1 neurons, forming synapses on the distalmost tips of the apical dendrites of area CA1. So I don't have a good picture of this. This is a dissected slice from the hippocampus, but here's area CA1, and here's the cell body, the basal dendrites here. And there is stratum radiatum, where the Schaefer collaterals come in and make synapses. And then there is also a layer that you can see with your naked eye in a very low-power dissecting microscope, where the axons coming from entorhinal cortex actually come in and make synapses in stratum lacunoso moleculare on these distal tips of the CA1 neurons. So we were interested in studying this pathway. Other groups had studied it and had come up with rather lackluster roles for these inputs, arguing that they could never make CA1 neurons fire. They were unclear what this input was really doing. It was largely inhibitory. So we wanted to study it more directly, hopefully optimizing the slice preparation. And to do this, we recorded extracellular field potentials in each of these layers. So we could record extracellularly in stratum lacunosa moleculare in response to stimulation of this input that comes from layer 3 of entorhinal cortex. And we could simultaneously monitor the field potential elicited by stimulation of the Schaefer collaterals. And then in some experiments, we could also record intracellularly from the cell bodies. And this just shows the kind of sink source or current source density analysis that you get when you stimulate the Schaefer collaterals and you record in stratum radiatum. You see a negative going field EPSP, and this is recorded as a tiny little positive going source in stratum lacunosa moleculare. This is how you know your electrodes are in the right place. In contrast, if we stimulate these, we call these temporoammonic fibers, first described by Cajal. If you stimulate those fibers and you record in stratum lacunosum, you also see a negative going field EPSP, and you would see in stratum radiatum a positivity. So in early studies actually that preceded most of what I'm going to talk about here, a graduate student in my lab was looking at how different patterns of activity, we were basically testing the hypothesis that going on the work of others, that in and of itself the temporoammonic input might not cause the CA1 neuron to fire, but we presumed that appropriately timed activity of these inputs could have an influence on whether information actually made it out of the hippocampus, since this is the last synapse. So the CA1 neurons are sort of the gatekeepers of information going out of the hippocampus. And what we discovered is that if one delivers either a single TA spike or a burst of TA spikes, and looks at either the ability of that TA input to either increase the probability that a spike, a Schaefer collateral stimulus is going to elicit a spike, or decrease the probability that a Schaefer collateral stimulus is going to elicit a spike. So here we have an SC stimulus that by itself only elicits a tiny little EPSP. And of course, if we deliver a burst of temporoammonic EPSPs, that can sum, and you can see now that the Schaefer collateral, that this normal little EPSP elicited by that stimulus now results in a spike that we can record at the cell body. And we characterized the interval over which this burst of temporoammonic inputs, which we delivered actually at the theta frequency, was effective. And we found that when the TA stimulus came on about 20 milliseconds prior to the 20 to 40 milliseconds prior to that single stimulus of the Schaefer collaterals, that gave us the greatest probability of eliciting a spike. And we also found the converse, and that is that if we increase the interval between the TA burst and that single pulse to the Schaefer collaterals, from this range now is 20 to 40 milliseconds. If we increase it to around 200 to 400 milliseconds, now what we find is that a stimulus that would normally elicit a spike, so we just turn up the stimulus current here, can be blocked, so that appropriately timed stimuli, a burst here delivered 200 milliseconds before, now blocks the spike. So you can see that the relative timing of the inputs is important. And of course, in this, we were thinking about the nature of bursting activity that would arise from CA3 and come directly to CA1 and then be meeting potentially the same kind of input that was delivered to the dentate gyrus. So we were trying to understand something of the temporal dynamics by how the timing of these temporoammonic bursts could influence information flow. So we could see very short intervals could potentiate spikes, and longer intervals could actually depress spikes. And this is actually because of the interneuron population that resides here in lacunosa moleculare, which serves as a potent, gives a big long IPSP to the the CA1 cells when you record from them. Now, we're also interested in whether not only spiking could be interfered with, but also plasticity. So if, for example, one is looking at long-term potentiation between the Schaefer collaterals and the CA1 cells, we developed protocols where we could, with a single Schaefer collateral stimulus, elicit LTP. And we showed that if we could repeatedly elicit LTP in this experiment. So we delivered a high-frequency train here. We could elicit LTP, and then we could just reset the baseline by turning down the stimulus, and we could. The LTP here was not saturated because when we delivered another tetanus, we could see more LTP. And we contrasted that with the situation where we delivered a burst of action potentials to the temporoharmonic input that was coincident with the LTP-inducing stimulus. And here we saw that this burst, this TA burst, could actually prevent LTP of the Schaefer collaterals. And if we then repeated the stimulus, just doing the Schaefer collaterals alone, we could see that we could elicit LTP. So this is an ensemble average, and this is a single experiment. And this shows the magnitude of potentiation that we see. And so in the experiments where we deliver both alone first, we see very little, actually no significant potentiation. And when we subsequently deliver a Schaefer collateral stimulus alone, we see that we can effectively elicit LTP here. And all of this is due to this strong inhibitory component that is produced by the temporoharmonic input, because if we do this experiment now in the presence of the GABA A receptor antagonist by cuculene, we completely prevent this inhibition of LTP, and actually we slightly increase the magnitude, which is a well-known effect of bicuculene at these synapses. Now, we also showed, and this was something that people had earlier discarded, that these synapses in and of themselves are modifiable, so that not only can one elicit LTP, but one can also elicit LTD. So this just shows a stimulation protocol that elicits, this is a Schaefer collateral pathway that is recorded at the same time as a temporoharmonic input, and a high-frequency train delivered here can elicit LTP, and this is blocked by an NMDA receptor antagonist. And not shown here is also, in another study, we showed that we could elicit LTD. So this pathway is modifiable, and it can modify the output and also the plasticity of the Schaefer collateral synapses. And in these experiments, we looked at whether plasticity, LTP or LTD of the temporoharmonic input modified the window that we had previously described for enhancing spikes or for blocking spikes. And basically what we found is that after LTP was elicited, so these are experiments where we assess the spike enhancing efficacy of a TA input before and after we elicit LTP. We find that after LTP is elicited, we see much more spike enhancement at these synapses. And after LTD, we see much less. So it's not a surprise, but it just shows that this pathway can modulate the output of the hippocampus and the plasticity, and that modulation itself is subject to plasticity at these own synapses. Now, after having done these experiments, and around the same time, actually, some work from the Mosers had shown, totally separate set of experiments, they had shown that lesions in CA3 appeared not to dramatically influence the place cell properties that they could record in CA1. And so, of course, those CA3 lesions would be predicted to interfere with that traditional trisynaptic circuit, but would be predicted to leave this direct input from the entorhinal cortex intact. And so we started thinking to ourselves that maybe it wasn't just that the temporoharmonic input might not be the modulator. Maybe it's actually conveying some of the information directly from the cortex to the hippocampus. And we even fantasized that maybe the trisynaptic circuit was the modulatory pathway and the temporoharmonic input was the real input. That's certainly a stretch. But so we decided to, and these were the sort of last days of the student who had done all the electrophysiology, we decided to tackle this, this topic directly, and not having time to do any fancy genetic silencing experiments and no channel rhodopsin on the scene, Miguel was actually trained as a vet before he came to my lab to do his PhD, and so we decided to look at animal memory in using the Morris water maze, which I'm sure you're all familiar with, where animals are placed in a submerged pool of water, and they must be motivated to get out of the water, and they get out of the water by finding a platform that's submerged below the surface so that they cannot see. And the only way they can find the platform is by bumping into it the first time, and then on subsequent trials, remembering the location of the platform relative to cues that are in the room. So it's a spatial memory task. And what Miguel did was to use electrolytic lesions to take out the temporomonic input here. So here's a hippocampal slice, and you can see this is where the fibers coming in from the entorhinal pathway would come in, and actually you can see in this dark area here, this is stratum lacunosa moleculare. So we did a series of experiments with targeted lesions, and then did behavioral experiments, and then after the behavioral experiments, sacked the animals and quantified the lesions, and then determined which animals were to be included in the temporomonic lesion group. And then there was also a group of sham animals that had the surgery and had the electrode lowered through the cortex, but it didn't penetrate the hippocampus. So the very first set of experiments we did were to perform these lesions, wait a period of time for the animal to recover, and then train the animal on the Morris water maze over a period of several days, and then do what's called the probe trial 24 hours after the last training. And in the probe trial, what happens is the platform is pulled out of the water, and you measure, as a metric of learning, how much time the animal spends swimming in the quadrant where the platform previously was. And so here for the sham-treated group, you can see T represents target. You can see that they spent a significant amount of time swimming in the location or the quadrant where the platform previously was. And in our temporomonic lesioned animals, we found, much to our disappointment, that these lesioned animals performed just as well as the control animals and spent a significant fraction of their time swimming in the vicinity of this platform. So these, as I said, were the last experiments that Miguel was going to do before he was leaving the lab. And after we had gone through this whole many months of optimization of the lesions and done this experiment, we were reluctant to just let it end there. So we decided to wait and look at long-term memory, having no idea at all whether the training protocol that we had used would be sufficient to sustain a long-term memory. So we waited essentially 4 weeks with this same group of animals, and we asked whether the sham lesioned animals could show a memory at 28 days. And that's shown here. So here's the same group of animals tested. That's their data that I just showed you. And this is the data shown for a probe trial conducted 4 weeks later. And we were happy to see that the control animals could still remember where the target platform was. And we're really surprised and also pleased to see that the temporomonic lesioned animals now perform significantly poorly when compared to the sham control animals. And this actually is not significantly different from these other groups. So somewhere in this, even though they showed perfectly good memory at 24 hours, sometime within that month, the absence of this input gave rise to a memory deficit that was recorded a month later. So what does this disruption represent? You can imagine that there's some sort of long-term memory process that's initiated during training that this input is required for. And this would be a process that would somehow be on a parallel track to the short-term memory that we assess with 24 hours. This is not unheard of in the animal learning literature. So it's possible that there's a long-term memory process that's initiated here that requires this input and simply fails because when we assess the memory at 28 days later, it wasn't present prior to training. Another possibility is that actually it's the temporomonic input that's required for this period of time after the animal is learned that's important for consolidating this memory. So maybe there is ongoing activity between entorhinal cortex coming into CA1 through these inputs that's required to consolidate this memory. To address this issue, what we did was to simply move the time of the lesion to after training, because if it's a long-term memory process that's initiated during training, we would predict that the animals would no longer show a deficit because the process would be initiated and started, and it wouldn't matter whether you have this input. By contrast, if this activity is required after learning, then we would expect to see a deficit in these animals that experience the lesion after the training trial. And so in this case, what we did was to train the animals, do a 24-hour probe trial, and then divide them into two groups randomly. So you can see here these two groups, the group of animals that were put into the sham control group and the group that were put into the future temporomonic lesion group, and they both show the same pattern of behavior with very strong memory for the target quadrant. These animals were then lesioned, and these animals had a sham lesion. They were put back into their cage, and then 28 days later we came back and assessed their memory. And what we found is that the sham lesioned animals, like the previous group, could still show memory for this quadrant, whereas the temporomonic lesioned animals showed no memory for the target quadrant. So that implied or showed that it was the activity after training that's coming in through this input that's actually required for the consolidation of this information. So this input comes from layer 3 entorhinal cortex to CA1. We call this the temporomonic input. And it modulates both the information transfer and the plasticity at the Schaefer collateral CA1 synapses. And these synapses themselves are plastic. They can be modified by both long-term potentiation and depression. And then in the behavioral studies, we showed that this activity on days following learning is required for long-term memory consolidation. So I think that this really points to, and there's a lot of data that support this, both in vitro data and also some in vivo data that really points to the importance of this long ignored input to the hippocampus, potentially in conveying information itself, but certainly in modulating information transfer out of the hippocampus and plasticity in the hippocampus. So all of the studies that I described using the human patients were done by Uli Ruteshauser as a graduate student and then as a postdoc. And Uli is actually now joined us in Frankfurt, where he's doing a postdoc in Gilles Laurent's lab, and he's also interested in continuing the work using human recordings. And then the animal behavior work on this input to the hippocampus was done by Miguel Ramondes. And these are the three surgeons that we've worked with. Adam Mamalak is my closest colleague. He's now at Cedars-Sinai. And there's also Ian Ross and Chris Heller. And these are the groups that we're grateful to for funding. And then lastly, just an unabashed advertisement for our new building. This is a rendering of the new Brain Research Institute that we're in the process of building. We are watching it being built. We've broken ground, and almost about up to here is built, the first floor. And there are 3 different departments. My department, Synaptic Plasticity, Gilles Laurent's department, Neural Systems and Coding, and then the department of Peter Mombart's Molecular Genetics. And then once the building goes up, we'll be searching for a 4th director. And then lastly, this is a rendering of the inside of the building. So we aim to build a very interactive and lively, high-energy institute for brain research. And I hope that we will see you there, and I hope I will see you here as well. So thank you.